Inside a film of water, a microscopic Milnesium tardigrade walks through moss using 8 short legs and tiny claws. Paired stylets pierce food cells, while a muscular pharynx draws liquid through the narrow mouth apparatus, within droplets.
Their transparent bodies contain complete digestive, nervous, muscular, and reproductive systems despite their extremely small size, within droplets.
Survival becomes difficult when the microscopic water film evaporates, and ordinary movement, feeding, and gas exchange become impossible. Instead of escaping across long distances, many tardigrades respond by transforming their entire physiological state before water vanishes. The animal withdraws its legs, shortens its body, and forms a compact barrel-shaped structure called a ton. Body contraction reduces exposed surface area while reorganizing tissues for severe dehydration and prolonged metabolic suppression during drying. At cellular scale, water leaves the cytoplasm and forces membranes, proteins, and organelles into increasingly crowded conditions, reversibly. Dissolved salts and metabolites become concentrated as dehydration removes the solvent supporting normal biochemical reactions. Without protection, crowded proteins can unfold, aggregate, or lose the precise shapes required for cellular function.
Tardigrade-specific intrinsically disordered proteins become important molecular protectants during drying.
Cytoplasmic abundant heat-soluble proteins can condense, form fibers, or create glass-like protection during dehydration, during drying. These proteins help stabilize biological structures while the cell passes through conditions that normally cause irreversible damage, reversibly. As water vanishes, protective molecules can create a vitrified state that greatly slows damaging molecular movement during drying. Vitrification does not freeze the cell like ordinary ice. It immobilizes components within an amorphous solid during drying. Laboratory experiments show tardigrade proteins can preserve enzymes and cellular materials during drying outside the animal, during drying. Small heat shock proteins add another protective layer by limiting aggregation and supporting protein quality control during drying. The visible result is a motionless ton containing living cells suspended near the boundary of measurable activity, reversibly. Androbiosis suppresses metabolism so deeply that ordinary signs of active animal life become extremely difficult to detect, reversibly. Energy use falls dramatically because the ton cannot replace fuel or remove waste through normal physiology during dormancy. Limited resources must preserve essential structures until water returns and full metabolism becomes possible again throughout cryptobiotic survival.
Drying can generate oxidative stress, making antioxidant systems essential.
Protection reduces damage, but revival also depends on repairing injuries accumulated during the inactive period throughout cryptobiotic survival. Many repair reactions cannot proceed efficiently until rehydration restores molecular movement and chemical transport. This conserves metabolic energy. Gradual environmental change can influence whether tardigrades enter and leave the ton successfully. This system conserves metabolic energy. Some species require slower drying to activate protective programs before cellular water disappears too quickly throughout cryptobiotic survival.
Desiccation tolerance varies widely, so one mechanism cannot describe every tardigrade lineage or developmental stage.
Trehalose contributes protection in some tardigrades, but many species produce little and depend on other molecular strategies, reversibly. Sugars and tardigrade proteins can cooperate, creating protective environments stronger than either component working alone throughout cryptobiotic survival. Water content determines whether protective mixtures remain fluid, form gels, assemble fibers, or become glassy solids during dormancy. Temperature and residual moisture strongly affect how long a vitrified biological system remains stable. This conserves metabolic energy. Desiccated tardigrades tolerate heat better than active animals, but survival declines with temperature and exposure duration during dormancy.
A temperature survived for minutes may become lethal when maintained for many hours. This network limits oxidative damage.
Cold tolerance follows different physiological routes, depending on whether the animal freezes while hydrated or already dry, reversibly. Cryobiosis suppresses activity during freezing, whereas anhydrobiosis begins by removing most body water. Different cryptobiotic states converge on profound metabolic suppression but begin through distinct environmental triggers. This coordinates rehydration recovery. Tardigrades can enter several reversible low-activity states, including responses to desiccation, freezing. Long-term survival requires preserving genetic information throughout every inactive interval and subsequent revival. This supports cryptobiotic survival. Ionizing radiation can break DNA directly or generate reactive molecules that attack genetic material indirectly during radiation recovery.
Radiation damage includes altered bases, single-strand breaks, and more dangerous double-strand breaks across chromosomes.
Antioxidant defenses limit secondary damage created when radiation interacts with water and cellular molecules. This supports DNA repair.
Surviving radiation requires both reducing initial injury and repairing lesions before normal cell division resumes during radiation recovery. Multiple repair pathways cooperate because no single system can correct every type of radiation-induced damage after irradiation. Some eutardigrades produce D-soup, a chromatin-associated protein that reduces damage under certain laboratory radiation conditions. D-soup represents one component within broader systems involving antioxidants, stress proteins, chromatin regulation, and DNA repair after irradiation. Radiation resistance evolved through diverse molecular combinations rather than one universal tardigrade gene. This program coordinates cellular recovery. Recent research maps coordinated molecular responses that activate after radiation damages cells. This network steadily stabilizes chromatin structure. Radiation exposure triggers extensive changes in gene expression and cellular metabolism. This coordinated program steadily coordinates cellular recovery. Some protection is constitutive, while other defenses activate only after damage begins. This pathway steadily supports DNA repair. Cellular quality control systems prevent damaged components from disrupting recovery. This framework steadily supports DNA repair during irradiation.
Survival of one adult does not guarantee normal reproduction or development afterward. This network steadily improves repair capacity.
Radiation can reduce fertility, hatching success, or later performance, even when exposed tardigrades revive. This protects genetic integrity.
To survive complete dehydration, the tardigrade enters anhydrobiosis, forming a protective ton. Dry tons often tolerate radiation differently because less cellular water remains available for producing reactive molecules after irradiation.
Radiation tolerance has limits, and sufficient damage eventually overwhelms protection and repair. This response steadily supports DNA repair. Successful revival means restoring coordinated cellular function, not merely surviving the exposure moment. This program limits radiation damage.
Freezing experiments reveal that hydration state strongly changes the physical stresses tardigrade cells experience. This stabilizes thermal recovery.
Removing water before freezing can reduce damaging ice formation inside and around cells. This pathway preserves molecular integrity.
Ice growth can deform tissues, concentrate solutes, and disrupt membranes during cooling and thawing. This limits exposure damage.
Cold survival depends on physiological preparation and controlled recovery rather than temperature alone. This response improves orbital tolerance.
Cooling rate can determine whether protective mechanisms activate before damage becomes irreversible. This network steadily coordinates post-flight recovery. Some tardigrades improve cold tolerance after experiencing moderate temperatures before severe freezing. This strategy steadily preserves molecular integrity. Thawing creates a second stress phase as concentrated solutes dilute and damaged structures reactivate. This preserves molecular integrity. Metabolic restart must provide energy without producing overwhelming oxidative damage. This program steadily coordinates post-flight recovery during freezing. Motor recovery usually follows rehydration or thawing rather than occurring instantly. This coordinated system steadily improves orbital tolerance. Vacuum exposure tests whether dehydrated animals can withstand extreme pressure loss without evaporating internal water throughout spaceflight recovery.
Drying before exposure greatly improves vacuum survival because little liquid remains available for rapid boiling throughout spaceflight recovery.
Active hydrated animals are far more vulnerable to vacuum than properly prepared tons. This process supports vacuum survival.
Vacuum is only one space hazard. Unfiltered solar ultraviolet can severely damage cells and DNA throughout spaceflight recovery.
Space exposure experiments found much better survival when samples were shielded from direct ultraviolet radiation throughout spaceflight recovery. In 2007, the TARDIS experiment exposed dried tardigrades to space conditions during a 12-day orbital mission. Experimentally, orbit combined pressure loss, cosmic radiation, and repeated heating and cooling. This coordinated program steadily supports vacuum survival.
Different exposure groups allowed researchers to separate the effects of vacuum and solar radiation. This preserves molecular integrity.
After recovery, scientists rehydrated the animals and measured movement, survival, and reproduction. This network steadily preserves molecular integrity. Some tardigrades survived space exposure, but direct solar ultraviolet sharply reduced success. This system steadily stabilizes thermal recovery.
A subset of survivors reproduced afterward, demonstrating recovery beyond temporary movement.
Post-exposure reproduction tests whether surviving adults retained enough cellular integrity for normal life history functions. After rehydration, long-term outcomes reveal damage that immediate revival alone can miss. This coordinated network steadily improves protein function. Ultraviolet radiation produces photochemical DNA damage that ordinary vacuum resistance cannot prevent. This response steadily improves protein function.
Recovery depends on lesion burden remaining within the animal's repair capacity. This coordinated response steadily enables research translation. Cosmic radiation differs from ultraviolet because energetic particles penetrate materials and create complex damage tracks during controlled recovery.
Tolerance combines physical protection during dormancy with extensive biological repair afterward. This framework steadily supports biological recovery. Surviving isolated laboratory stresses does not prove tardigrades could establish populations on Mars. This process preserves sample stability. Active growth and reproduction still require ordinary biological resources unavailable across most extraterrestrial surfaces. This maintains molecular function. Cryptobiosis preserves an existing organism. It does not create a self-sustaining ecosystem. This program stabilizes cellular integrity. Tardigrade experiments inform discussions about biological transport while leaving natural interplanetary transfer unproven. This network enables research translation. A complete transfer scenario includes stresses far beyond simple orbital vacuum exposure. This strategy steadily stabilizes cellular integrity. Experimental survivability cannot be converted directly into probability for natural panspermia. This coordinated response steadily supports biological recovery. Their greatest scientific value lies in revealing general principles of cellular protection and recovery. This preserves sample stability. Purified tardigrade proteins can protect enzymes, membranes, and other biological materials during controlled drying. This improves protein function.
EAHS proteins can adopt different material states depending on hydration, concentration, and surrounding molecules. This preserves sample stability.
Versatility allows protection during stress without permanently trapping cellular machinery after recovery. This framework steadily enables research translation.
Researchers explore tardigrade proteins as tools for preserving sensitive biological products.
Unprotected proteins retain function better than untreated controls after desiccation. This coordinated strategy steadily enables research translation.
Promising experiments have not yet produced universal preservation technology. This coordinated network steadily preserves sample stability during preservation.
Tardigrade survival emerges from coordinated systems rather than one miraculous molecule. This coordinated process steadily maintains molecular function. Engineered protection must work across different molecules, cell types, and drying conditions before practical applications become reliable, carefully.
Tardigrade-inspired systems may stabilize selected biological materials without reproducing the entire animal's cryptobiotic physiology. Reducing dependence on refrigeration could improve storage and transport in places where cold chains remain difficult during research.
Laboratory stabilization studies suggest possible routes toward more resilient biological products.
Safety, dosage, manufacturing, and regulatory questions must be solved before clinical use. This process steadily clarifies biological understanding. Dehydration protection research may also support agriculture, biotechnology, and long-term sample storage. This strategy preserves scientific accuracy. Tardigrade mechanisms offer additional strategies rather than replacing every existing preservation technique. This response steadily clarifies biological understanding. Each method balances cost, damage risk, storage duration, and recovery requirements. This coordinated strategy steadily supports research design. Dry preservation could reduce logistical burdens during long missions if protected materials remain functional. This preserves scientific accuracy. Tardigrade biology does not provide a pathway for placing people into reversible cryptobiosis. This process limits exaggerated conclusions. Human organs require continuous circulation, neural coordination, and temperature control that tardigrade tons temporarily suspend throughout future research. Scaling protective mechanisms across biological organization creates increasingly difficult engineering problems. This coordinated strategy steadily preserves scientific accuracy. Tardigrades diversified across many habitats, producing different combinations of tolerance and life history traits. This guides realistic interpretation. Extreme survival occupies only part of a normal life cycle centered on growth, feeding, and reproduction during research. Developmental stage can strongly influence tolerance, recovery, and reproductive consequences. This pathway steadily clarifies biological understanding during evolution. How tardigrades enter anhydrobiosis. The outcome expands into humidity, temperature, drying rate, duration, and species variables surrounding one tardigrade ton. Tardigrades are extraordinarily resilient yet vulnerable to sufficient heat, radiation, pressure, dehydration, or prolonged damage throughout future research. Their defining achievement is reversible shutdown, not immunity to physical limits. This coordinated system steadily strengthens future experiments. Recovery transforms a motionless ton back into a feeding, moving animal. This coordinated program steadily clarifies biological understanding. Tardigrades survive extremes by protecting cellular structure, suppressing metabolism, and repairing damage when favorable conditions finally return, carefully.
This small lizard escapes predators by diving underwater and repeatedly breathing from a silver bubble attached to its snout.
Water anoles inhabit tropical stream margins, where dense vegetation, wet rocks, and moving water provide immediate underwater refuges.
Their streamside habitat places branches, roots, and climbing surfaces directly above pools, channels, and concealed submerged shelter.
Perching close to water lets the animal disappear from danger within seconds, before many terrestrial hunters can react.
Birds, snakes, and other predators create intense pressure for rapid escape behavior along exposed rainforest stream banks. Before diving, the lizard evaluates distance, cover, current, and the nearest safe route beneath the water safely.
A sudden leap carries the animal below the surface before most land-based predators can follow or accurately track it.
Once submerged, depth, darkness, ripples, and visual distortion make the small reptile surprisingly difficult to locate from above.
Strong toes and curved claws help it remain anchored against flowing water while hiding beneath rocks or roots.
The extraordinary respiratory behavior begins when air leaves the nostrils and forms a bubble instead of floating away. Hydrophobic scales help exhaled air remain connected to the face, keeping the bubble accessible beside both nostrils. Semi-aquatic anoles possess scale features associated with stronger water repellency and more reliable underwater bubble retention. The nasal bubble may connect with a broader reflective air layer trapped against hydrophobic scales across the head.
Air moves through the nostrils while the mouth stays closed, preserving a compact respiratory circuit beneath the surface.
The lizard inhales from the bubble, exhales back into it, then repeats the same cycle during submersion.
Every breathing cycle changes the bubble's composition because metabolism consumes oxygen while tissues continuously produce carbon dioxide.
Oxygen follows the ordinary vertebrate route from the lungs across thin tissue and into the circulating bloodstream.
Rebreathing extends the usefulness of existing lung air, rather than creating an entirely new respiratory organ underwater. Measured oxygen decline inside the bubble confirms that its trapped air actively supports the submerged animal's respiration. Repeated inhalation lets the lizard reuse air that would otherwise escape immediately toward the stream surface after exhalation. Recycling stretches the useful duration of a limited air store, while the animal remains concealed during a defensive dive. Each extra underwater minute increases the chance that a searching predator leaves before the lizard must resurface. Staying below the surface allows currents, shadows, and ripples to erase the original location of the sudden escape. Recorded submersions vary greatly because body size, stress, temperature, habitat structure, and individual decisions all influence duration. Some semi-aquatic anoles have remained underwater for roughly 8 minutes, although most defensive dives are considerably shorter. A maximum observation reveals biological capacity, not the routine dive length of every animal under natural conditions.
Body size, water temperature, current speed, and disturbance can all change how long a submerged lizard remains hidden.
Smaller and larger animals carry different lung volumes, metabolic demands, and bubble surface areas relative to body mass. Remaining nearly motionless lowers oxygen consumption, allowing the bubble's limited respiratory value to last substantially longer under threat. Sheltered positions behind roots and stones reduce physical effort while hiding the lizard from movement above the water. Calm pockets behind obstacles demand less energy than exposed current, where the animal must constantly resist downstream force.
Moderate flow can refresh gases around the bubble,
without tearing the delicate air layer away from the snout. Carbon dioxide readily dissolves into water, creating a possible route for respiratory waste to leave the bubble. Because carbon dioxide is highly soluble, surrounding water may absorb it faster than oxygen disappears from the reservoir. An exposed bubble water boundary could therefore remain useful longer than a completely sealed pocket containing the same air.
Metabolic carbon dioxide travels from active tissues through blood toward the lungs, even while the lizard remains submerged. Exhaled carbon dioxide enters the nasal bubble, where some molecules may diffuse outward into the passing stream. Carbon dioxide removal is physiologically plausible, but recycled stored air remains the mechanism supported most directly by experiments.
Underwater rebreathing combines ordinary lungs, hydrophobic skin, trapped air, and a dynamic gas boundary beside the nostrils. The respiratory loop begins with oxygen already stored inside air, carried beneath the surface during the initial dive.
Oxygen concentration falls predictably while a living lizard continues breathing repeatedly from the attached reservoir underwater.
Researchers compared that decline with artificial bubbles held underwater under similar physical conditions, but without breathing animals.
Control bubbles retained oxygen more consistently, demonstrating that biological respiration caused the stronger decline seen with lizards. Those measurements confirmed that the nasal bubble functions as an extension of the animal's existing pulmonary air supply. Oxygen enters the lungs during inhalation, while mostly unused nitrogen returns to the bubble during the following exhalation. As oxygen decreases, nitrogen occupies a greater fraction of the remaining bubble, and the respiratory mixture becomes progressively poorer.
Eventually, available oxygen falls below a useful level and contributes to the lizard's decision to return toward air.
The animal releases its underwater grip, swims upward, and breaks the surface to obtain a completely fresh breath.
At the surface, the depleted bubble merges with open air, while the lungs refill within a single respiratory cycle. After climbing onto a nearby rock, the lizard restores normal oxygen delivery while monitoring the streamside environment for danger. Heart rate, breathing, and movement gradually return toward ordinary levels as the animal recovers between defensive dives.
Another predator may appear before recovery finishes, forcing the lizard to repeat its underwater escape under greater physiological pressure.
A second dive creates another nasal bubble immediately, showing that the behavior is repeatable whenever danger returns.
Every resurfacing restores a fresh gas mixture, replacing the oxygen-poor reservoir produced during the previous submerged interval. Across a single day, repeated cycles of diving, rebreathing, resurfacing, and recovery may occur around the same stream refuge.
Underwater movement may also support exploration or foraging, although predator avoidance remains the best established ecological function. Submerged stones, roots, and leaf pockets contain potential prey, as well as concealed resting places away from terrestrial hunters.
The lizard balances underwater opportunity against limited oxygen, physical effort, and the continuing need to return for atmospheric air.
Scientists tested the bubble's actual respiratory value by experimentally preventing its normal formation during controlled defensive dives.
Hatched lizards entered identical water chambers, differing mainly in whether the familiar nasal air bubble remained available.
Control animals preserved their ordinary hydrophobic air interface and continued cycling exhaled gas beside the nostrils underwater.
A temporary surface treatment disrupted bubble attachment without changing the chamber's water temperature, depth, or overall handling conditions. After treatment, exhaled bubbles detached from the face and floated upward before the lizard could inhale them again.
Those animals relied mainly on their initial lung supply, because reusable gas no longer remained beside the nostrils.
Lizards retaining normal bubbles stayed submerged substantially longer than animals whose external air interface had been disrupted. In that experiment, ordinary rebreathing increased average dive duration by approximately 32% under otherwise matched conditions.
Comparable body sizes, chambers, and handling procedures strengthen the conclusion that access to the bubble caused the difference.
The result demonstrated that the air pocket performs a functional respiratory role, rather than merely reflecting trapped surface air.
However, the experiment did not show permanent underwater breathing, unlimited oxygen renewal, or complete independence from atmospheric air.
The bubble remains a finite reservoir whose volume and gas composition continue changing throughout every submerged breathing cycle. Cycling improves how the initial air supply is used, but it cannot prevent eventual oxygen depletion during long dives. Possible carbon dioxide loss across the bubble surface may further delay the point when resurfacing becomes physiologically necessary. Gas may move in both directions across the interface, although each proposed exchange process requires separate experimental evidence. Confirmed rebreathing, plausible carbon dioxide removal, and emerging oxygen uptake evidence therefore occupy different levels of scientific certainty. Successive studies progressed from behavioral observation toward gas measurement, bubble disruption, and controlled environmental experiments over several years. Natural dives remain essential because laboratory mechanisms evolved to solve ecological problems along predator-filled tropical streams.
In the wild, bubble-bearing lizards hide beneath ledges and roots, while threats continue searching along the bank above.
An extended dive allows danger to move farther away before the concealed animal cautiously returns to the surface. Comparative research revealed that underwater bubble rebreathing appeared independently in several distantly related semi-aquatic animal lineages. Separate evolutionary branches developed remarkably similar diving behavior, despite lacking one recent bubble-breathing common ancestor within Anolis. Streamside species from different American regions encountered comparable combinations of predators, water access, and underwater shelter near flowing water. Those recurring ecological pressures repeatedly favored escape strategies that turned nearby streams into temporary defensive refuges during attacks. Across independent lineages, threatened lizards entered water and formed accessible nasal bubbles using similar behavioral sequences under threat. Ordinary animal skin already possessed water-repellent microstructure before specialized underwater rebreathing evolved within semi-aquatic species. Natural selection likely co-opted that existing surface property, giving hydrophobic scales an additional respiratory function underwater. Water repellency may originally support self-cleaning or surface protection, while later becoming useful for stable bubble retention. Semi-aquatic forms maintain more persistent external air films that remain attached during movement beneath the water surface. The accessible air concentrates near the nostrils, allowing repeated inhalation while the lizard keeps its mouth closed underwater.
Different species vary in bubble size, attachment area, head shape, and the number of threads within their air layer.
Convergent solutions still reflect each species' body dimensions, scale texture, habitat, and preferred underwater hiding behavior underwater.
Local stream conditions can determine which bubble geometry and diving strategy produce the greatest survival advantage locally.
Fast rocky channels and slow shaded pools impose different costs through current strength, oxygen availability, and concealment. Calm water reduces the energy needed to hold position, though surrounding gases may refresh differently than in strong flow.
Water containing more dissolved oxygen creates stronger conditions for oxygen to move toward an attached underwater bubble.
Researchers therefore compared dive duration across chambers containing low, intermediate, and high dissolved oxygen concentrations in controlled trials.
Lizards remained submerged longest in the highest oxygen treatment, when water movement was kept relatively low and controlled.
Separate experiments manipulated dissolved oxygen and flow independently, preventing one environmental variable from masking the other's effect.
With oxygen held constant, slower flow treatments also produced longer dives than faster-moving water inside the experimental chambers.
The combination of adequate dissolved oxygen, low physical effort, and a stable bubble suggested an additional respiratory possibility.
A true physical gill would draw some dissolved oxygen from surrounding water into the bubble during submersion.
Aquatic spiders and insects already use air films or bubbles that exchange respiratory gases directly with water.
Across such interfaces, oxygen can enter from water, while carbon dioxide leaves through the same exposed boundary.
The animal bubble changes volume and shape during dives, potentially recording several simultaneous gas movements across time.
Closed reservoir and physical gill models predict different oxygen trajectories, bubble volumes, and achievable underwater durations for the animal. Longer dives in oxygen-rich water match one important prediction expected from partial physical gill activity underwater. However, Researchers have not yet measured continuous oxygen flux directly across the bubble's surface during natural rebreathing. Future microsensors could monitor gas movement at the interface without disturbing the lizard's behavior or hydrophobic air layer.
Simultaneous oxygen, carbon dioxide, and volume measurements would reveal how storage, waste removal, and replenishment interact underwater.
Portable field sensors can connect controlled respiratory mechanisms with temperature, flow, and dissolved oxygen in occupied streams. Wild lizards frequently perch near stream sections offering manageable current, adequate oxygen, and nearby underwater cover during danger.
Roots, ledges, and cavities provide better concealment than open banks, even when respiratory conditions are otherwise similar.
Seasonal rainfall, sediment, temperature, and vegetation can alter both the quality of refuges and bubble performance throughout the year. Warmer streams hold less dissolved oxygen while simultaneously increasing the lizard's metabolic demand during underwater hiding at the same time. Shaded riparian forest may preserve cooler, oxygen-rich channels better than exposed streams altered by canopy loss. Bubble breathing, therefore, depends on an intact combination of stream structure, water chemistry, vegetation, and predator ecology. Repeated breathing, oxygen decline, bubble removal effects, and dissolved oxygen experiments now support several connected parts of the mechanism. Evidence is strongest for recycled air, while direct oxygen replenishment from water remains an active research question. A tiny nasal bubble helps this lizard stay hidden by combining stored air, gas exchange, and remarkable surface biology.
This seabird can sleep while remaining airborne above the open ocean. Long flights inspired a question that behavior alone could not answer. Researchers studied breeding female great frigate birds from a Galapagos colony. A lightweight recorder was attached before each bird returned to flight.
Electrodes measured electrical activity from both brain hemispheres.
An accelerometer recorded head motion and turning forces.
A separate GPS unit tracked position and altitude.
Together, the instruments linked brain state with real flight behavior.
Some recorded journeys covered thousands of kilometers without landing. The longest sleep recordings followed birds aloft for about 10 days. Over deep water, the birds had nowhere safe to perch.
Frigate birds avoid prolonged contact with the ocean surface.
Their plumage is less waterproof than that of many seabirds.
Getting airborne after becoming soaked would be difficult. Low wing loading makes sustained soaring unusually efficient.
A deeply forked tail provides precise aerial control. Atmospheric updrafts supply altitude without constant flapping. Circling converts rising air into height. Stored altitude then powers long descending glides.
Soaring reduces the muscular cost of staying airborne.
Direct brain recordings finally revealed whether flight contained genuine sleep. Sometimes one hemisphere entered slow-wave sleep, while the other remained more awake.
Bird vision is strongly crossed between eye and opposite hemisphere.
The awake hemisphere could monitor visual information from the open eye. During asymmetric sleep, The open eye often faced the direction of turning. Visual monitoring may help avoid collisions inside crowded rising air. Half-brain sleep preserved some vigilance during flight. Hemispheric sleep also helps other birds monitor danger on land. In frigate birds, vigilance appears linked to navigation and nearby traffic.
The hemispheres can differ in sleep depth, rather than switching completely.
Sleep exists along a continuum of hemispheric asymmetry. Frigate birds also slept with both hemispheres simultaneously. Bihemispheric sleep showed that one awake hemisphere was not always required.
Aerodynamic stability can persist through short bouts of whole brain sleep.
Updrafts reduce the need for active altitude control.
Most flight sleep occurred while birds circled in rising air.
Flight provides time before a sleeping bird approaches the water.
Birds generally remained awake near the ocean surface. Foraging demands favored wakefulness during daylight. During flight, that half-brain sleep allowed rest without surrendering all visual attention. Eye state matched the turning direction during many asymmetric sleep bouts. Either hemisphere could become the deeper sleeping side. Sleep arrived in short fragments rather than long, continuous periods. Typical flight sleep bouts lasted only around 12 seconds. The longest uninterrupted episode remained under 6 minutes. Dozens of brief naps accumulated across darkness. Sleep occupied only a small fraction of time aloft. Recorded birds slept during less than 3% of flight.
Daily flight sleep averaged less than 1 hour.
Ecological demands kept the birds awake most of each day and night. Frigate birds track ocean features associated with future feeding opportunities. Following changing ocean patterns may require sustained attention. Large predators can push prey toward the surface. Frigate birds capture food while remaining airborne.
They sometimes harass other seabirds for food.
Kleptoparasitism provides another aerial feeding strategy.
Efficient feeding must support days of continuous movement. Aerodynamics minimize the cost of remaining aloft. Night flight involved even less flapping than daytime travel.
Despite sleeping in flight, the birds obtained remarkably little sleep.
Land sleep exceeded flight sleep by more than an order of magnitude. Back at the colony, sleep became longer and deeper. Land allowed more continuous slow-wave sleep.
Flight sleep was generally lighter than land sleep.
A secure surface removed the need for aerodynamic control.
The recorded females were returning to chicks between foraging trips.
Parental demand made long foraging journeys necessary.
Food resources could lie thousands of kilometers away.
Returning to land created an opportunity for extended sleep.
Researchers still debate how completely land sleep repays losses from flight. Available recordings do not capture every transition between sea and land. Longer studies could measure cumulative sleep recovery.
New biologgers may reveal how sleep changes during longer flights.
Tracking studies show longer airborne journeys than current brain recordings cover.
Continuous flight does not automatically prove continuous in-flight sleep.
Brain activity is required to identify sleep reliably. EEG distinguishes the major avian brain states.
Rare flight bouts included rapid eye movement sleep.
Brief REM did not visibly disrupt the recorded trajectory.
Avian REM episodes can last only seconds. Reduced muscle tone was brief enough for flight to continue. Atmospheric support may buffer momentary reductions in active control. Several safeguards overlap during these rare episodes.
REM sleep ended rapidly. Most sleep aloft consisted of slow-wave sleep.
Both asymmetric and symmetric patterns occurred.
Sleep strongly favored circling flight.
Tension remained highest near potential feeding zones.
Birds often gained height before sleeping. Flight sleep combined several strategies rather than one simple autopilot. Altitude reduced immediate danger from descent.
Thermals supplied passive support.
Unihemispheric sleep preserved directional awareness. Brief naps limited exposure to reduced attention.
Together, these features made sleep aloft possible.
Human responses cannot be inferred directly from a seabird. How frigatebirds function on so little sleep remains unresolved.
Several physiological explanations are still under investigation.
Sleep may be regulated more locally than whole brain labels suggest. On land, the bird finally gains a stable place to rest.
Both hemispheres can enter deeper sleep. Daily sleep rises to more than 12 hours. Slow wave activity becomes stronger. Adhika.
Sleep episodes become more continuous. REM sleep also occurs safely on land.
Resting conditions permit fuller sleep architecture. Land sleep likely contributes to recovery. Complete repayment of lost sleep remains uncertain. Future studies may reveal the hidden costs of oceanic sleeplessness. Other species may use different sleep strategies. Long flight alone cannot establish sleep on the wing.
Smaller instruments could expand direct tests.
New recordings may reveal multiple solutions to sleep during migration. Great frigate birds remain the first birds directly proven to sleep in flight. Their recordings revealed several unexpected sleep states. One eye can monitor the turn while the opposite hemisphere rests.
Sometimes the entire brain sleeps while flight continues.
Yet total sleep remains astonishingly small at sea. Frigate birds sleep in the sky by combining brief naps, soaring, and partial vigilance.
Above powerful thunderstorms, enormous electrical flashes can appear where the atmosphere is already approaching the edge of space. These events occur far above ordinary weather, across atmospheric layers most people never associate with lightning. Above storms, conventional lightning moves through clouds or toward ground, but other electrical disturbances illuminate the sky overhead, across altitude. At those heights, air density, pressure, and electrical breakdown behave very differently from conditions near Earth's surface. Thin air allows enormous luminous structures to develop from surprisingly small amounts of excited gas, above active storms. Excited nitrogen produces much of the characteristic red light seen in high-altitude sprites. Structure identifies mesospheric response. Most transient luminous events are faint, brief, and easiest to detect with sensitive cameras after darkness falls. A complete event may unfold within milliseconds, disappearing before an observer can consciously process its shape. Across altitude, high-speed imaging transforms one mysterious flash into a sequence of rapidly developing electrical structures. During brief flashes, the storm below can trigger light tens of kilometers above its cloud tops without a continuous visible bolt. Understanding the upper flash begins with how collisions inside thunderstorms separate electrical charge. This signal resolves storm charge. Violent collisions among ice and water particles help organize charge throughout a growing storm. Contrast tracks mesospheric response. Storm dynamics create large reservoirs of opposite charge separated across several kilometers of cloud. Boundary locates lightning coupling. As charge separation increases, the surrounding electric field strengthens throughout the cloud and nearby atmosphere during brief flashes. Lightning begins when electrical breakdown creates conductive channels through air that was previously insulating. Transition compares electrical breakdown.
Some sprite-producing flashes transfer positive charge from the cloud toward Earth in exceptionally powerful strokes. After lightning,
That rapidly changing field can push thin upper air beyond its local breakdown threshold. Emission exposes mesospheric response.
Red sprites occupy the mesosphere and lower ionosphere, dramatically higher than thunderclouds or commercial aircraft, within thin air.
Their scale becomes clearer when placed above aircraft routes and below most orbiting spacecraft. Evidence frames sprite formation. A sprite can illuminate a region far larger than the storm feature that appears to trigger it. Enormous size does not imply long duration, because most visible sprite structure develops almost instantly, during brief flashes. Some sprites follow their parent stroke almost immediately, while others appear after a measurable delay, above active storms.
Both rapid charge removal and continuing current can influence whether the upper atmosphere reaches breakdown within thin air.
Researchers use charge moment change to describe how strongly lightning alters the electric field above a storm.
Charge moment changes strongly increase the probability that a lightning flash produces a sprite across wide regions. Even powerful positive strokes do not guarantee sprites, because atmospheric conductivity and storm geometry also matter within plasma. Sprite initiation depends on several interacting conditions, rather than one simple lightning measurement. This boundary maps global occurrence. Nighttime conditions favor optical detection and change the electrical response of thin mesospheric air above active thunderstorms.
Solar radiation alters electron density, affecting how electric fields build and dissipate above storms. Contrast tracks charge transfer.
Many events are observed at night because their faint emissions would disappear against bright daylight across wide regions.
Observers often photograph sprites from distant clear skies rather than standing beneath the parent thunderstorm during brief flashes.
Distance can improve visibility by exposing the entire region above a storm's cloud shield. Transition compares global occurrence.
Sensitive imaging captures emissions too brief and dim for ordinary cameras under most conditions. Network supports upper fields.
Precise timing links each upper atmospheric event with candidate lightning activity inside the storm below, after powerful lightning. These matched observations establish the connection between sprites and powerful lightning charge transfers. This channel distinguishes storm geometry. Sprites occur worldwide wherever sufficiently energetic thunderstorms interact with favorable upper atmospheric conditions. They are not limited to tropical convection, although storm structures and triggering patterns can differ above active storms. Many sprites resemble luminous jellyfish, with a bright body suspended above long descending tendrils. Channel tests ionospheric change. Individual streamers propagate through localized electric fields and build the sprite's intricate lower structure. Evidence frames radio effects. Ionization grows around moving streamer tips, producing visible channels that divide repeatedly. This observed response measures ionospheric change. Different streamer polarities can develop in opposite directions within the same enormous event. This observation reveals halo development. Other events appear as simple luminous columns with little branching visible at ordinary video speed, within thin air. Sprites frequently occur in clusters distributed across a broad region above the parent storm. Observation records ionospheric change.
Successive sprites can appear to dance across the sky as lightning activity moves through the storm, across altitude.
Wind shear and tilted storm charge can shift sprites away from the parent ground stroke during brief flashes. The upper discharge may not sit directly above the point where lightning reaches the ground, above active storms.
A broad sprite halo sometimes appears before or without bright streamer development. This synchronized geometry reveals halo development.
Halos arise from diffuse electrical excitation rather than the narrow channels defining mature sprite streamers after powerful lightning.
Under suitable fields, a halo can transition into structured streamers that grow downward rapidly. Boundary locates ionospheric change.
Microsecond imaging reveals a developmental sequence hidden inside the brief visible flash. This measured sequence links streamer growth. Bright streamers can travel extraordinarily fast through the thin upper atmosphere. This field explains streamer growth above storms. The event extinguishes once local fields can no longer sustain ionization and energetic electron collisions within thin air. Although the light vanishes quickly, the upper atmosphere can remain electrically disturbed afterward. This sequence connects plasma motion.
Sprites and related events can produce measurable changes in very low frequency radio propagation. Emission exposes streamer growth.
Remote radio measurements help detect electrical disturbances even when clouds or distance block optical observations during brief flashes. Combining cameras, radio sensors, and lightning networks reveals how thunderstorms couple electrically with the upper atmosphere above them. Blue jets begin near storm tops and propagate upward through the stratosphere, rather than appearing suddenly far above. Unlike sprites, blue jets develop upward from the thunderstorm itself. This signal explains jet development within thin air. Typical blue jets terminate well below the highest sprite structures. This transition compares event rarity within thin air. Different colors reflect atmospheric density, composition, and the energies exciting nitrogen molecules. This mapped profile connects charge transport. Blue jet emissions arise from excited and ionized nitrogen within denser stratospheric air above thunderstorm cloud tops. Ion streamers combine into the luminous structure recorded by sensitive high-speed cameras, revealing jet development frame by frame. Observed blue jets follow extremely brief electrical activity embedded within the cloud. This profile links plasma structure. Storm charge arrangement determines whether breakdown remains internal or escapes toward the stratosphere. This pattern tests global circuitry. Some upward discharges grow beyond ordinary blue jet altitudes and enter a different category. Response measures cloud escape. Gigantic jets can create a direct electrical connection between thunderstorms and the lower ionosphere. Profile connects jet development. Their upper branches reach approximately the same height as the base of the ionosphere. Pattern distinguishes event rarity. A single gigantic jet can extend many times higher than the storm producing it. Contrast tracks plasma structure. High-speed observations show that gigantic jets can advance through repeated leader steps and streamer bursts. Across altitude, different plasma structures coexist within the same upward discharge. This response constrains cloud escape within the upper atmosphere. Most attempted branches terminate before reaching the ionosphere. This profile links jet development within the upper atmosphere. The discharge changes structure as air density and conductivity vary with altitude. This mapped evidence frames charge transport.
Observed gigantic jets can transfer substantial electrical charge directly upward. This response measures global circuitry within thin air.
Gigantic jets represent one visible pathway connecting storm electricity with the planet-wide atmospheric circuit across wide regions. These jets are uncommon relative to the enormous number of lightning flashes occurring worldwide. Trace constrains global circuitry. Limited observations still prevent a complete global climatology of gigantic jets, especially across remote tropical oceans and continents. Elves are enormous, faint rings produced when a lightning electromagnetic pulse strikes the lower ionosphere, within thin air. An ELVE can spread across a region hundreds of kilometers wide within a fraction of a millisecond. The parent lightning does not need to send a physical channel into the ring. Measurement maps historical discovery.
Electromagnetic energy briefly excites the lower ionosphere and creates the expanding optical emission. This observation reveals historical discovery.
Its geometry reflects the outward-moving electromagnetic pulse intersecting a thin atmospheric layer. This timing exposes sensor fusion. Most ELVs vanish within about a millisecond, making them among the fastest transient luminous events within thin air.
Different event families occupy distinct timescales despite all being associated with thunderstorms. This synchronized boundary locates optical detection.
Photometers aboard spacecraft are especially effective at detecting ELVs. This sequence links sensor fusion within thin air. Viewing from above removes cloud obstruction and reveals the event's true horizontal scale. This contrast frames ELV expansion. Repeated orbital observations help estimate occurrence rates beyond what ground cameras can measure. This boundary maps historical discovery.
The first confirmed sprite recording appeared accidentally during an unrelated nighttime camera test in July 1989.
Repeated viewing revealed that the strange flash was real rather than a camera artifact. Contrast tracks event timing.
Shuttle observations soon provided more examples and confirmed that the phenomenon extended across large storm systems, across altitude. Different perspectives revealed structures that looked confusing or incomplete from the ground. This brief sequence links event timing. Coordinated observations reconstruct altitude, size, and timing more accurately than any single camera. Researchers combine optical imagery with storm structure and electrical measurements to identify triggering conditions. Network supports sensor fusion.
Millisecond synchronization separates plausible parent flashes from unrelated lightning occurring nearby. This structure supports ELV expansion above storms.
Raster cameras expose plasma motion that ordinary video compresses into one featureless flash. This channel distinguishes optical detection. Spectroscopy identifies which molecular transitions generate each event's characteristic light. This pattern distinguishes historical discovery within thin air. Color and spectrum provide remote clues about otherwise inaccessible upper atmospheric conditions. This map structure identifies event timing. Space-based instruments now watch thunderstorms from above, where cloud tops cannot hide upward electrical activity after lightning.
ASIM combines imaging, fast light measurements, and high-energy detectors during repeated passes over active storms, above storms.
Orbital measurements captured blue jets beginning with extremely brief electrical activity inside cloud tops. Measurement maps forecast skill. Multi-channel observations distinguish genuine events from reflections, cosmic rays, and ordinary lightning glare. Field resolves ionospheric response.
From orbit, ELVs appear as complete rings rather than partial arcs hidden near the horizon, above active storms.
Astronaut photography complements automated instruments by targeting storms selected from forecasts and real-time observation. Within thin air,
Successful observations require predicting where intense storms will intersect a spacecraft's narrow viewing window. Observation records remaining uncertainty. Skilled citizen observers now contribute valuable images, timestamps, and storm locations to scientific databases. Timing clarifies sensor coordination. Multiple viewpoints can estimate altitude and structure, even when no dedicated instrument observed the event. During brief flashes,
reconstructions reveal how upward leaders develop inside storms before escaping into the atmosphere. Profile connects ionospheric